Genetics is the study of the patterns in how genes are inherited or passed on through the generations. When we studied meiosis and how the chromosomes lined up at the metaphase plate randomly, that this caused their egg or sperm to have a different mix of alleles than the mix they were given at fertilization of their parents' egg and sperm. And that this mix is so important to genetic variation in the population. It creates genetically unique individuals. They have the same genes, of course, but they have a different mix of alleles than their parents had. Again, because of meiosis that produced their parents' sex cells. So here's a name you should know, Mendel. He figured out with experiments some of the rules of inheritance. His test subject was the garden pea. Now, Mendel knew nothing about meiosis. Microscopes were just being developed at that time. So he didn't know anything about DNA, anything about chromosomes during meiosis. So all he had to work with were traits. Okay, like in pea plants, the color of the pea that was produced, the texture of the pea, was it wrinkled or smooth, the tallness, the height of the plant, the maximum height it grew to, etc. Now, pea plants was, as I said, his experimental organism or his model organism, and it was convenient to use this because they grow quickly. You don't have to wait years for them to mature and then have offspring. And they produce a lot of offspring in a short time, so you can see patterns in the offspring being peas. Also, Mendel, with this pea plant, he could control the matings. Okay, so let's talk a little bit about the flower. A flower has, the petals, but in the middle it has this vase-like thing. And inside that vase-like thing, there's the eggs. That's the female part. Then there's stamens. And the stamens have anthers at the end where the pollen forms. Okay, so pollen is the male, is like the sperm, the male gamete. And then inside the ovules of, this is called the pistil. Inside the ovules, or little compartments inside the pistil, are the eggs. And look, that's the female gamete. And the male things, these stalky things are called stamens. And the stamens, the end thing is called the anther, but you don't need to know that yet. Okay, the point is, when a pollen grain gets stuck on the tip of the pistil, it will grow a tube down through the neck of the pistil and until it finds an egg, and then it will fertilize that egg, and that egg will become an embryo, and it'll be protected in a seed coat eventually. We call that seed. And then that pistil, the bottom part called the ovary, will develop into a pea pod with a bunch of peas in it. Okay, so he could control who fertilized who by cross-cutting out the stamens and then brushing on the pollen from another type of pea plant, same species but different traits, and see what the offspring would turn out as. So he cross-pollinated different pea plants of the same species but with different alleles by dusting the pollen from that plant onto another individual plant. Okay, so here you see how Mendel did his experiments. Like, here's a purple flower plant, here's a white flower plant, pea plant, different traits, petal color. So here's the pistil, and he cut off the stamens. He cut off the stamens so that the plant would not self-fertilize. And he went to another plant with a different trait, like white flowers, and he used a little paintbrush to brush off the pollen and transfer it to the stigma, the sticky part of the pistil, so that he controlled these who mated with who this way. Okay, so here's a generic picture of a flower. Stamens, I already said. The stamen, you should know this, the stamens produce the pollen. They're the male part. The female part, I call the pistil, but they call it the carpel. That's the part that produces the eggs. The stigma of the pistil or carpel is where the pollen sticks on. Okay, the pollen is produced in the anthers. They get all powdery with pollen. Meiosis occurs in the anther, and to produce sperm, and meiosis occurs in the ovule, in the pistils. We call that the ovary, just like we would a woman's place where the eggs are formed. Okay, so meiosis occurs in the little rooms called the ovule, where the eggs are. And those eggs will produce, once fertilized, they'll produce the pea pods. they'll produce the seeds. In this case, peas. Now, the ovary will turn into the fruit. In this case, the pea pod that protects the seeds. So this guy came from the ovary of the pistil or carpel, and these guys came from their seeds. They come from the ovules inside the ovary. Of the pistil. Basically the eggs. So here are the traits that Mendel had to work with his pea plants. So we had either round or wrinkly seeds. We had yellow peas, which are seeds, or green seeds. We had white flowers or violet flowers. We had full pods or crinkled pods. We had yellow pods or green pea pods. We had the flowers where the branching occurs, axial flowers. We have flowers on the tips of the branches, terminal flowers. We had plants that grew tall and plants that only stay short. All these things are traits. You either have one or the other. These are distinct traits. You call them characteristics. We call them traits. More scientifically, we call these phenotypes. The phenotype is the trait that you can observe. It's what it looks like. Now, we know that the trait is due to the gene that codes for that trait. Now, a trait is simply the proteins, due to the proteins that make up that trait. Okay, for example, you have brown eyes, or blue eyes, or hazel eyes. That color of your eye is due to a protein, and that protein is coded for by an allele for eye color, the gene for eye color. There's different forms of that gene that we talked about called alleles. Now, Mendel knew nothing about genes. All he could measure and understand were the traits. Now, we call that trait that you can observe the phenotype. The allele that codes for that trait is the genotype. And of course, Mendel didn't know anything about that, but we'll be using that term. The genotype is the allele or the gene form that codes for that trait. The code being the part of the DNA that has instructions for making that protein that is what the trait is based on. True breeding. Let's get used to some of the terms we use today. True breed means whenever you have an individual with certain traits and you cross it with another individual with the same kind of traits, you'll always get children, offspring, that have the same traits. Okay, so that is that is true breeding. So in this case, we have yellow seed individual crossed with a yellow seed individual. You'll always get in the offspring, every pea will have yellow, will be yellow. In this case, this is true breeding for green peas. Okay, so he isolated purebreds. This also be called a purebred. For yellow seeds, but we say that purebred is true breeding. Every time you breed it with an individual that with yellow seeds, it will produce children that have yellow seeds. Okay, he used these terms. Okay, so this is the parental group, the parental generation. His original experiment. Now, in this case, he's crossbreeding. He's crossing. Let's just say crossing. He's crossing a green pea plant with a yellow pea plant. So what was it? I mean, he's taken the pollen from the yellow seed plant and brushing it on the pistils of the green seed plant flowers, or vice versa. And then he's seeing what the offspring look like. Okay, offspring would be what the children turned out to be. Now, in this cross, by the way, that's called the first filial generation. Just think of it as first family. Okay, like the kids. So, in this case, what happened was you don't get a mixture of green and yellow peas. You got all yellow peas, no green peas at all in the first F1 generation. That's kind of weird. What happened? Then what he did was he took two of those, he cross-pollinated them. Okay, remember, these are the children of cross between a green and a yellow seed plant. Okay, so he took two of the yellow seed children. All the children were yellow seeded. And he got, we're going to find out, he got some green peas, individuals, and some yellow pea individuals in the next generation, the F2 generation. And he found that there was a ratio there of 1/4 of the kids were green, and then 3/4 were yellow. 3/4 of the children were yellow pea. So this is what he did, and we'll explain why that happens like that. He didn't really know why, but he was just observing it. But now that we know genes, we can explain it. So, a monohybrid cross is where the parents differ only in one trait. That's where you get mono from. Let's do a flower color one. So the parent generation was purple flowers. He cross-bred them with white flowers. All the plants in the first first generation had purple flowers when you grew their seed, the seeds from this cross. No, he caused these flowers to self-fertilize. So he took F1 generation and these guys are not purebreds. They're called hybrids because they have a mixture of the purple and the white in them. Now, if you self-fertilize those F1 plants, those hybrids, what happens is, like I said, you get 3/4 being one of the traits and 1/4 of the children having the other trait. So this is a 3-to-1 ratio, phenotypic ratio. So the phenotypic ratio of the F2 generation is 3-to-1. Purple phenotype to white phenotype, white flowers. And he did this with all kinds of traits. So something hidden, the white flower trait was hidden, phenotype was hidden in the F1 generation, but it came out in the F2 generation. So what Mendel found, he said that there were heritable units. We call them alleles. Well, we call them genes, but there's different forms of that gene. But he had no idea what a gene was, but he inferred it from his experiments. Okay, so there's two versions of heritable unit. We call them alleles. There's the white flower and the purple flower. The purple flower was masking the white flower color gene in the F1 plant. So F1 plant carried the two alleles for the flower color gene, but it didn't show the white flower phenotype. So for all the other traits that I talked about that Mendel studied, he kept on finding the same pattern in the F1 generation after he self-crossed the hybrids. Okay, so we talked about alleles. Alleles are different forms of a gene. For example, purple or white flowers are based on two different alleles for the same gene, the flower color gene. Okay, so I shouldn't really say that white and purple are alleles. White and purple are the phenotypes based on the alleles for white and purple flowers in the flower color gene. Okay, so we said that one of the genes is masking the other gene in the F1 generation. So we saw that white flowers was masked. The white gene was masked by the purple flower gene. Actually, I should say allele. So the way we express this is white is recessive to purple. Or we could say purple is dominant. Or we can just say white is recessive, the purple gene is allele is dominant. Oh, so I don't get too picky. I could say I'm going to just be brief and say white is recessive, purple is dominant. We would use letters to represent the alleles. So usually you take the dominant letter as the capital one. So P, let's make a little base on it to make it look like a big P, is the allele for purple flowers. And that little p, that's supposed to be a little p, is the allele, lowercase p, for white flowers. Now, there's 3 different combinations you can have. You can have big P, 2 alleles. An individual always has 2 alleles. A gamete only has 1, because an individual organism is 2N, whereas a gamete, pollen or an egg, is haploid. Sperm or an egg is haploid. So big P represents what we call homozygous dominant genotype. Now, I'm showing the genes, I'm showing the alleles. So this is a homozygous dominant genotype. What's the phenotype of 2 capital P's? This would be a purple phenotype, purple flower phenotype. Now, if you had little p, lowercase p's, this would be homozygous. The genotype would be homozygous recessive, because they're both the ones that get masked. But the phenotype would be white flower phenotype. Now, you could have a hybrid, like in the F1 generation. Big P, little p. This would be called a heterozygous genotype. And because the big P masked the little p, this would be also a purple phenotype. So when you have a purple flower, you don't know whether it's big P, little p genotype or big P genotype. So this would be the hybrid. It's really important that you distinguish between phenotypes and genotypes. Genotypes are the 2 alleles. And what homozygous, recessive, homozygous, dominant, and heterozygous mean. Here they just review what dominant versus recessive alleles are. I just did that. Okay, so what they're saying here is that a sex cell carries only one allele for each trait. That's because during meiosis, when the sex cell was formed, the germ cell went to haploid, so you only have half the alleles you had when you were diploid. So when we set up a Punnett square, you probably did this in grade school. You set up a Punnett square, for example, big P, little p, times big P, little p. Up here will be, it doesn't matter who you do it. Let's say the dad, and right here's the mom's. The dad's sex cell will either have a big P in it or a little p in it. And the mom's sex cell will either have a big P in it or a little p in it, because meiosis separates alleles. You either have one or the other. You won't have both. But then the kids that form will be in these boxes down here in the Punnett square. So that's due to fertilization. This dividing up of the alleles is due to meiosis. So you'd have big P, big P, big P, little p. You'd have big P, little p, and then little p, little p. So it crosses the phenotypic ratios. Okay, this would be the F1 cross. This would be the F2 generation. This would be an F1 cross of the two hybrids. Okay, so the phenotypic ratio in the offspring would be what? 3 purples to 1 white. Right there, that 1 white one. Genotypic ratios would be 1 big P, 1 homozygous dominant to 2 heterozyguses to 1 homozygous recessive, right? Just look in the boxes. So Mendel found this before we knew anything about chromosomes, DNA, meiosis. So when we did see it under a microscope, meiosis and mitosis, it all clicked together. What Mendel was finding made sense with what we were seeing under the microscope. And that what he called the hereditary factors, we now call genes or alleles. They're located on the chromosomes. We already talked about this. So a 2N cell, a diploid cell, has, let's say, your skin cell. It has 2 sets of every chromosome. 1 came from your mom's egg, and 1 came from your dad's sperm. And that in your zygote, and the zygote just did mitosis to form you. Okay, so we call the locus, the location of a particular gene on a chromosome. Um, so each genetic locus individual has two alleles, one on each homologous chromosome. So here's a locus, maybe for eye color. Here's a locus for hair texture. Okay, let's say S is straight hair. Little s is curly hair. So let's say mom had curly hair and dad had straight hair. So I'll put the alleles like that. And then let's say dad had blue eyes, which is recessive, and mom had brown eyes. So mom had big B and dad had little b at that locus. Okay, now these are one chromatic chromosomes because, I could have shown them like this. Each one has an X, right? But they're making it simple. So these would be what the chromosomes would look like right after mitosis, before they get sister chromatids. Okay, so this would be a 2N cell. Now, a gamete would just have either the purple or the red one. It wouldn't have both because that would be N haploid. Okay, let's do some problems. A plant with purple flowers is allowed to self-pollinate. Generation after generation of self-pollinating, it keeps on producing only purple flowers. This is an example of true breeding, forming purebreds. Children only. So what makes pea plants a good subject in Mendel's experiments? Peas show easily observed variation in a number of characters such as, they have different traits, that's good. It is possible to completely control matings, yes. Like I told you, when he cut off the stamens and brushed on pollen from another plant, so we have more than two things. It's possible to obtain large numbers of progeny from it. That is true. Peas have unusually long generation time. No. That is my choice, because that is not. Pea plants have a very short generation time. You plant the seed, boom, in a couple weeks it grows to a tall, mature plant and is forming seeds. Okay, so which of the following was not a reason why pea plants were good? And that would be letter D. Genetic crossing in which a parental organism is different. Only one trait is known as monohybrid cross. Alternate versions of a gene are called alleles. An organism has two identical alleles for a gene is said to be homozygous for that gene. And the organism is called a homozygote. I didn't really say that, but I didn't say that second part, but That should make sense. If you are homozygous for a gene, that means you're a homozygote for that gene. That doesn't mean you're a zygote. We're now going to talk about Punnett squares. I kind of brought those up already. Okay, here again we're reviewing the different kinds of genotypes. The lowercase letter represents the allele for the recessive allele, and uppercase letter represents the dominant allele. Homozygotes could be either homozygous dominant, two dominant alleles, or homozygous recessive, two recessive alleles. If you have a combination of dominant and recessive, that's called a heterozygote or a heterozygous genotype. We usually put the dominant first, so don't write it like that. Little a, big A. I already did this with you guys. What if you get a cross between purple flower, purebred purple flower, and a purebred white flower? So you set up one of the parents on top of the boxes. You just split it down the middle. That's all. That indicates what happens during meiosis to make the haploid gamete. And you write the letters on top and the letters for the other mate down the side. Then you just put them together. Put them together. I always put the capital letter first. Okay, so the genotypic, in this case, all the kids. Okay, so this would be the parental cross. What I just showed you was the F1 generation. F1 generation is all heterozygotes. So we get, for the F1, we get 100% heterozygous genotype, or 100% purple. Okay, so this would be the first one would be the genotypic ratios, and the second one would be what it looks like, the phenotypic ratio. And here you see, that's what they got. And what will be the distribution of flower colors of these? 100% purple, because big P is dominant to the little p allele. So the only way you can have white flowers is to not have the big P there. Big P be there. So what am I giving you here? I'm giving you here the phenotypic ratios. And right here, the genotypic ratios, 100% are heterozygous. Okay, so now we're going to take the F1 generation and we're going to cross it. So I'm going to take one of those individuals and cross it with itself. So big P, little p, big P, little p, big P, big P, big P, little p, big P, little p, little p, little p, So now now we have, it's supposed to be a P, little p. So again, would be the genotypic ratios in the F2 general from this F1 cross. The F2 kids will have a genotypic ratio in the offspring of 1. Let's do it with words instead of symbols. 1 homozygous dominant. I'll be homodom. To 2 heterozygosis. To 1 homorecessive. And in those F2 generation, the phenotypic ratios would be 3 purples, right? To 1 white. Recall that P is, the big P is the allele for purple flower. And little p is the allele for white flowers. Okay, instead of using the 1 to 2 to 1 ratio, you could say 1/4 to 1/2 to 1/4 if you want. Or you could say 25%, 50%, 25% like they did. So whatever way you should be able to understand and do if I have a multiple choice question. Okay, but the phenotypic ratios, this is the genotypic ratios, right? But the phenotypic ratio would be 75% to 25%. 75% purple, as I said down there. Okay, in pea plants, tallness is dominant to shortness. So we would use big T and little t as the allele letters. Big T is tall, little t is short. Now, they'll give you this when they give you a problem. Okay, if you cross a homozygous tall, okay, so that'd be, homo means the same, times a heterozygous tall. That'd be a mixture. What percentage will be tall? So they're asking phenotypic ratio. So I'm just going to do one parent, split them up. That's showing how the alleles separate during meiosis in that parent sex cell. And this showing how the alleles separate in that parent sex cell. So do the cross. These go together. And look what happens. I get all of them being tall. All of them being tall because big T is dominant. So my genotypic ratio is different than my phenotypic ratio. My phenotypic ratio is what they're asking, 100% tall. My genotypic ratio, which they didn't ask, is 50% homozygous tall. Homo dominant and 50% hetero. So there's the answer to verify what we just did. Here's another question. Yellow seed is dominant to green seed. So heterozygous yellow seed plant. Okay, so that'd be big Y, little y. Is crossed with another heterozygous yellow seed plant. So big Y, little y. So big Y, big Y, big Y, little y, big Y, little y, little y, little y. What percentage of offspring will have yellow seeds? Okay, it looks like 75%. 3 out of 4, which is 75%. So you could say 3/4 of the offspring. You could say 75% if you want, because you have to have a dominant allele to show the yellow color, because what they said here. And so that's showing you that we're correct. Okay, so if you had big P, and let's say big P stands for purple, and little p stands for white. Looks like pink. Would you be able to tell phenotypically the difference between these two? A homozygous dominant and a heterozygous? No. Both the heterozygous and the homozygous dominant have purple flowers. So, how would Mendel be able to tell the difference between these? Or how would you be able to tell whether one is purebred or one is a hybrid? Well, you do what is called a test cross. A test cross is a cross that will tell you which one of these is. The evidence of the offspring will tell you whether it's homozygous, dominant, or heterozygous. So what you do in a test cross is always cross it with the unknown with homozygous recessive. Always. So when you do a cross, look, they make these Punnett squares kind of at a diagonal, which is kind of weird. So when they cross big B with little p, you'll get these ratios. When you do big P, little p, with a cross little p, you'll get these ratios. So if the parents are going to be homozygous dominant, all the kids are going to be pink. 100% of the kids will be pink, phenotypically. And then if it's heterozygous, It's heterozygous, like right here. All the kids are going to be, no, 50% of the kids will be white, and 50% of the kids will be pink, purple. I should be saying purple. So you don't really have to know the genotype to figure out the genotype, because you can tell by the cross results when you cross it with a homozygous recessive what the genotype would be of the purple flower plant. Okay, this is very important to understand. This is called a test cross. Again, a test cross is taking something that is expressing the dominant allele. You're wondering, well, is it purebred dominant or is it heterozygous? So you do a cross with a homozygous recessive. If you get any individuals in offspring that have the recessive trait, the white flowers in this case, then you know that the parent was heterozygous. But if you only get the dominant trait in the offspring, then you know that the parent was homozygous dominant. So let's do some review questions. Organism with two identical alleles, that's homozygous. And then he would be called a homozygote. The homozygous genotype makes him a homozygote for that gene. Okay. This would be yellow. I guess little y would be green if we're talking about pea plants. A plant with yellow seeds could have which of these genotypes? Well, like I just said, all you have to do is have one big Y in your genotype. So it could be big Y, or big Y, little y. So you could be either one of those, and you'd have to do a test cross like we just talked about to figure out which one it is. Okay, two true breeding parental plants with these genotypes for seed color. I guess big Y. Okay, big Y means yellow. Little y means green. Seeds. The F1 hybrids will have the genome. Okay, so we did big Y times little y, and we get obviously you get all of them being heterozygous. So they would all be yellow, but so, and they would all be big Y, little y genotype. Okay, the tall plant has a genotype big T. The only genotype of the egg or sperm could be, one could be big T, the other has to be big T. So remember, meiosis separates the two N to N, and because you're homozygous, the only allele you have for this tallness gene is big T. So egg or sperm has to have big T. Okay, how about same question? What is the genotype of the egg or sperm? It could be big T or little t. Can't be two letters together. So letter B is the right choice. Okay, what color would the F1 plants be in a cross between big P, little p, little p? Okay, it looks like, you're going to start memorizing this probably. Looks like every single plant will be purple. So why are all the F1 plants purple when true bred purple and white plants are crossed? Okay, I just did the cross. All of them are going to be big P, little p. Why is that? Because every possible genotype of the offspring has a dominant allele in it. So the best choice would be A. You might look at D and say, well, all the pea plants have purple flowers. Well, that's true, but why? Why do they all have purple? So that doesn't really answer the question. So, red is dominant, white is recessive. So what's the probability of having white flowers? So you'd have to be R, R, little r, little r. So let's do the cross. Okay, put the parents, split up the parents into individual alleles because that's what happens during meiosis. Then I do the Punnett square. I see that there's no way I'm going to get little r, so 0%. 100% chance that you'll be red flowers. Okay, so this one I should know by now. It's a 3 to 1 ratio, 75%, 21%. But let's just keep on doing it. Okay, the probability these plants will produce offspring with red flowers. Okay, remember, big R is dominant. So that would be 75%, right? Not 25%. If you have a big R anywhere, it's going to be dominant, red. So 75% red flowers, 25% white flowers. Okay, I'd have to give you that. You don't have to memorize which one's dominant. So white is recessive. So the F2 generation, so F1 was produced, just remind you, F1 was big P times little p, two purebreds. And that produced 100% big P, little p in F2. Then we took the F2 generation and crossed that. And you should know by now, I'll do the cross. That would be a big P, big P, little p, big P, little p, little p. So that'd be a 75% to 25% ratio of purple to white, or a 3-to-1 ratio of purple to white. Ding. An organism's physical traits are known as its phenotype. Red is dominant over white. Okay, so parent A. Well, parent A is, I look at this, and parent A has homozygous, is a heterozygous, but red is dominant, so he has red flowers. I'm assuming that this is complete dominance. Now, some traits are incomplete dominance, as we'll find out coming up, and so it could be pink, but it says is completely dominant. I assume it's completely dominant, not incompletely dominant. Red is dominant over white, completely, I guess. Parent B has red flowers too. Plants below can produce a plant with red flowers. Yeah, all their plants will be produced have red flowers. True, because you got a big R here, and you got at least one big R here, so sure it can. Now, just to show you, all the plants will produce red flowers. Only way you can produce white flowers is both parents have a little R, which they don't. So I'm doing a long way of solving this, but that's not necessary. The probability that the parent plants A and B will produce red flowers, I just showed you 100%. If both, the only way you could produce a white flower is if the other parent was little r, or big R, little r. You have to have both parents having a little r to be able to produce a white flower. So 100% red flowers. White flowers, I just told you, you'd have to have a little R here. So there's 0%. I did a cross in the previous problem. Okay, so two hybrids. So again, I'd have to tell you purple is capital P dominant, white is little p. So he crossed two hybrids, which would be like that. Again, big P, big P, big P, little p, big P, little p, little p, little p. Okay, so again, that would be purple to white. You got 75% purple, 25% white, or 3 out of 4 to 1 out of 4 ratio. So it's that one. 3 purple to 1 white. Okay, the expressed physical traits again is phenotype. We did that one. Homozygous recessive. Okay, so this is a test cross. You're taking an unknown genotype that has, okay, so that's like taking something like this and crossing it with homozygous recessive. And depending on whether this is A or little a, you can determine which one the big A question mark is by the test cross. Whether an allele is dominant or recessive depends on how common the allele is. No, it has nothing to do with it. No. Okay, so whether it or another determines the phenotype. Okay, so that would be it. I don't really like how it's worded. It doesn't depend on what chromosome. So the next step Mendel did was, with his traits, was to do crosses with two traits being different in appearance. This is called a dihybrid cross. So instead of focusing just on one trait, you want to see if the law of segregation would be exhibited with two traits. That is, would, if you had like two plants that were crossed, but one had different color of flowers than the other and different color seeds than the other, with these two genes, he called them factors, with these two factors work, do the same Punnett square kind of thing with independent of each other, or would one affect the other and change the expected ratios. So I'll show you actual example in a minute, but what if you had like A, B, B, okay, times A, B, B, two different traits. And they were heterozygous for both. That'd be like the F1 generation bit by me crossing A, B, B times A, B, B. That'd be the parental. That'd be the parental cross. Two hybrid. I'm sorry, two purebreds. This would be the F1. The result of that would be the F1 generation. This would make all A, B, B kids. And then you take those kids and you self-cross them. And you're going to get a 9:3:3:1 ratio. 9 dominant in everything. That would be A. Well, this is genotype. These are these are phenotypes. So you get 9 that were big A anything, big B anything. You get only 1 out of the 16. These add up to 16. You get only 1 out of 16 that are A, little b, little b, little a, little a, little b, little b. You get 3 out of 16 that were dominant in the first trait and recessive in the second. And you get 3 out of 16 that are recessive in the first trait and dominant in the second trait. Now, when I put a little slash here, that means it could be big B or little b. I should have told you that. So you could remember 9:3:3:1 ratio instead of doing the whole thing out. They should have given you a little thing here, a little legend here box. So yellow pods. So little g is yellow pods, the pea pods, and big G is the green pea pods. And big Y is green seeds. I don't know why they, wait. So yellow peas are dominant. To green peas, which would be recessive, would be okay. I'm figuring it out from there. I would tell you with a menu like that. Okay, so we have a cross like this. Okay, so we're going to do a Punnett square with G, G, Y, Y times big G, little y. Now, this is time consuming in a way. I'll show you a shortcut after I talk about the long way of doing it. Now, what you do is FOIL this in order to figure out what goes in the Punnett square top and side. So you FOIL this. Okay, so first, outer, inner, last. So first, I get little g, little y. Outer, I get little g, little y. Inner, I get little g, little y. Last, I get little g, little y. So I could have just saved time and said all those are going to form little g, little y gametes. Okay, and this one, I can just do the same thing. They're all going to be big G. I'm going to take a shortcut. They're all going to be big G, little y. So when these two are the same, you know that the only gametes that will form will be little g, then big Y. So I just transfer that thinking here. So really it's just one cross like this. So for offspring, you get big G, little g, big Y, little y. I'll keep the G's together and the Y's together. So you're going to get 100% of the offspring being that. Okay, you can see here that all this stuff I crossed out is repetitious. And this right here is what we did, right? So all the plants would be have green pods and yellow seeds, the same phenotype. Now, what if we cross those? What if we cross these guys? It gets a little more complicated. Set up a Punnett square. So know it's going to be, okay, let's do, they're both the same size, so we don't really have to. So what we do on this, we'll do over here as well, because they're both the same genotype. So let's split this up, the FOIL method. First, outer, inner, and then last. So the first is big G, big Y. The second is outer, big G, little y. The third, inner, is big G, little y. No, little G, little. God, why can't little G, big Y. And the last is little G, little y. Okay, so I could repeat this because they're both the same. I'll be sure you're making the letters really different, or else you get really confused. Okay, then I put it together, keep the G's together and the Y's together. This is really time consuming. I'll show you a shortcut in a few minutes. I would not do this on a test because it takes way too long. I'll probably mess up. I'm trying to do this fast. Notice I always put the capitals first. Okay, so I'm gonna get 9:3:3:1 ratio. Let's just verify that. So we expect 9 out of 16. There's 16 possibilities. 9 out of 16 will be big G, anything, big Y, anything. I'm doing the genotypic ratios. Okay, so 3 out of 16 will be little g, big Y, any Y. 3 out of 16, we'll flip it around, will be big G, anything with 2 little Y's. And only 1 out of 16 will be everything little. Okay, so that would be the genotypic ratios. And I could do the phenotypic ratios as well. So they did it all for you here. they're doing the actual genotypes, the actual genotypic ratios for each specific time. I'm not about to do that. So the whole point is you only get 1 out of 16 being with yellow pods and green seeds. And all the rest, and then you get 3 out of 16 with green pods and yellow seeds. And then you get, where's the little y, Then you get 3 out of 16 being green pods and green pods and green seeds. You can count them all. I'm not going to do that. So it's really the phenotypic ratio, which is the one I'll ask about on the test. So you get 9-3-3-1 ratio with 9 having both dominant traits expressed, 3 having the dominant trait on 1 expressed and a recessive trait on the other allele expressed. And then flip it around, 3 out of 16 having the recessive allele expressed in the other trait and the dominant allele expressed in the other one. Then both recessive traits would be 1 out of 16. That's the one you should know. So the law of independent assortment, Mendel stated that genes separate independently of each other. Okay, I'm going to show you a shortcut way that's really powerful, so you don't have to deal with the Punnett squares on these dihybrid, trihybrid, tetrahybrid crosses or more. So there's 2 rules, and the only rule I use is the multiplication rule. So the multiplication rule states that the probability that 2 or more events will occur is a product of their individual probabilities. So that's all I want to know from this. For example, what if I ask you, what would be the chance that a plant would have both yellow seeds and yellow pods? Okay, so in a cross like this, what was it? Now, here's the legend telling you for pod color and pea color. So on this cross, how could you have both yellow seeds and yellow pods? So we're looking at yellow seeds. So if you have yellow seeds, it would be big Y, or big Y, little y, right? So I'm going to just put big Y slash. And also I want what? Yellow seeds, yellow pods. Yellow pods would have to be 2 little g's, because you're not going to get yellow unless you have 2 recessive genes together. So I take my cross and I do the Y's only. Now I know this is 3 out of 16 if you memorized it, and if you started with those 2 ones, those 2 parents. But let's just prove it a shortcut way. So I take the y's and I cross them. What I get is big Y, big Y. I'll just be quiet and do it. Okay, so what's the chance of me getting big Y something? That'd be 3 out of 4 chance of getting big Y with something next to it. So that'd be 3/4 here. Okay, now I'm going to do the same thing with to find what the ratio would be for little g, little g. Okay, so that I take those two guys and do the same thing, even though it's kind of ridiculous because I just did it with the y's. So the only way I can get little g together would be 1 out of 4 chances. So that'd be 1 out of 4 ratio. Now, the multiplication rule says multiply the two probabilities to get the total probability when you have two things happening at the same time. So it's 3/16. And that's what I memorized maybe up here. So it checks out. Okay, so we have two independent chances, two independent events. Okay, so what's the chance that both will land heads up? So what's the chance of one ending lands up 1/2, right? Then you multiply it by the chance of the other landing heads up. So you'll get 1/4 of a probability that both will land heads up. Okay, so you could have memorized this. So 1 out of 16 will be little g, little g, little p, little p. Mendel stated, this is a law independent of independent assortment. Will be separated into the gametes independently of each other. The law of segregation just allows you to let's say we had a genome like that allows you to say that those alleles will be segregated or separated into the gametes individually. You don't get both of them at the same time. So doing that from this allows you to use the law of segregation. But the answer would be for two independent ones, letter B. Okay, organism heterozygous for two characteristics. Well, even if it, so something like this, well, he'd be a hybrid for each one, so he'd be a dihybrid. Because there's two. Now, I really need to do some more of these problems. What if you had a trihybrid like this? And then you crossed it with something like that. Okay, what is the chance of getting, or the probability of getting, an offspring like this. A, B, B, and then let's say D, D. Okay, so what you have to do is use just the A's, do a cross with those, A times A, and then you do a cross with just these guys. Then you do a cross with these guys. Instead of doing a really complicated one. This is easy if you do that law of probability multiplication rule. Okay, anyway, let's do that one. A. So you get half and half. So what's the chance of me getting little a from this cross? 1/2. So let's write that. 2 out of 4. 1/2. Okay, let's do the same thing with this one. Okay, so I'm doing genotypic ratios, okay? So big B times big B, little b. Okay, so what's the chance of getting big B, big B? That looks like half again. Half a chance right here. So that'd be I'll put 1/2 right there. What's the chance of getting big D, little d? So do a Punnett square. So that'd be half as well. So what's the chance of me getting this? That would be 1/2 times 1/2 times 1 over 2 times 2 times 2 is 1/8. 1/8 of the kids will be that. With the same cross, what if I asked you, what's the chance of getting little a, little b, little b, little d, little d? Well, let's do the same thing. Little a is 1/2. I mean, I did the work already up there. Little b, ooh, no chance at all, 0. Little d, 1/2. 1/2 times 0 times 1/2 is 0. 0% chance of getting that. Let's do another one. C. Let's not use C. Q times S times T. Let's do a cross with Q S and T. Kind of like a test cross. Okay, So what's the chance of getting a child that's, let's say, big Q, anything, big S, anything, then TT? Okay, so let's do individual crosses for each of these. So QQ, do a cross. So the chance of getting big Q, anything, is 1/2. These two out of 4. Okay, how about SS times little SS times S little s? What's the chance of getting a big S in your answer? Well, 4 out of 4. So I'll just say times 1, or 4 out of 4, 100%. So the chance of that is 1. Chance of this is 1/2. Then the chance of getting big little t out of big T, little t times little t is 1/2, it looks like. So my total chances are 1/2 times 1/2 times 1 is 1/4. So 25% of the kids will be like